Good morning, everyone. So I decided to make a class about the process of photosynthesis. So if you guys may have forgotten, it is the process whereby plants make food for themselves. So if you guys would like to follow along in your books, it's in your textbook from page 121 to 141. But anyway, later on you guys will have to go through it anyway in the book, but that's the work. We're covering all 20 pages of that. So but let's get back to photosynthesis. What is it about? It's an anabolic process, which means the entire process uses chemical reactions, which makes small molecules, take small molecules and turn it into bigger molecules, in this case, food for the plant. So what it needs is sunlight, is the energy needed for photosynthesis. The first fact it needs, sunlight. Then it needs carbon dioxide. And thirdly, it needs water to sustain it. Then to enable the process of photosynthesis, we need the light, as mentioned, the chlorophyll. The chlorophyll is the part, the green pigments inside of the plant cells in the chloroplasts, where it is the process happens. And then it also needs enzymes. So enzymes are a kind of catalyst, and catalysts are things that speed up the reaction process, or in general reaction processes. So in this case, the enzyme speeds up photosynthesis. And then your end products is sugar, or usually in the form of glucose. That is your sugar product, the food, and then also produces oxygen. It's your byproduct. Okay, moving on. So photosynthesis can be broken down into two phases, your light and your dark phases. Okay, don't misunderstand, light and dark phases does not mean that the light phase happens in the day and the dark phase happens at night. It's not the case for the dark phase. In the sense of the light phase would be this side. The light phase, yes, should happen during the day because we need sunlight. But the dark phase of the entire cycle does not necessarily happen in the nighttime. It also can happen during the daytime. The phase just means it does not need sunlight, because by this time energy has already been made inside the chloroplast, and then that energy, which is known as ATP, is used within the dark phase. Okay, just sidetracking a bit. Everything here you see is within the chloroplast. So this is the structure of the chloroplast, wherein the chlorophyll lies. So this entire process of photosynthesis happens within these structures, which are known as the thylakoid, and each little round disc is known as a granum. Okay, so the chlorophyll is within these little structures. You can see what they look like on the inside. Cross-section through the inside is known as the lumen. Okay, so heading back to the light phase. So what happens during the light phase? We need water, and we get oxygen. Okay, so light goes in. We need light and we need water for this reaction to happen within the thylakoid, and the byproduct is water is released. Okay, so then as soon as energy has been produced during the light phase, known as ATP or NADPH, it's your, let's say it's the batteries of the plant. The batteries have been recharged, and now this energy of the batteries is being used during the dark phase. The dark phase cycle is known as the Kelvin cycle. We're not going into much detail about the Kelvin cycle, but during this time in the dark phase, we need carbon dioxide, and sugar is produced. The food is produced in this stage. Okay, so since the batteries, the energy of the batteries have been depleted here, the energy has been used. NADP and ADP, discharged batteries, we can call it. This goes back to the light phase to gain more energy, and as soon as the energy has gained, as soon as it's gained energy, it turns back into ATP and NADPH. Okay, looking into more detail into the ATP thing. Okay, I'm calling it charged batteries because that's the energy source. It's not really a battery. But here it shows ATP means it's charged, or you have energy, and ATP, that form, means it does not have energy. Okay, so ATP and ADP, they are molecules. Okay, you have adenosine. Adenosine is a type of nucleic acid attached to a ribose. This is a sugar, and this sugar is attached to three, in this case, phosphate groups. And these, the bonds that keep the phosphate groups together, that is what gives us the energy. Okay, so this is ATP, adenosine triphosphate. So this is the 1, 2, 3 triphosphate groups. Okay, just for you guys to understand it. So when the energy is used, what happens? One of these phosphate groups, it goes away. Okay, and as soon as this bond, the bond between these two break, we get energy. And that is what's being used during these chemical reactions. Okay, so one is lost. Now it's adenosine diphosphate. Di, 1, 2. We lost that one. So now the energy is gone, but then as soon as we gain more energy, it goes back being a triphosphate. Okay, that's basically on a molecular level what happens. So what do we use, or what does the plant use this energy for? Well, in plants and humans, it's used to make macromolecules. So macromolecules like your proteins, fats, and nucleic acids. So it's used to make these molecules. Also, in muscles, specifically in humans and animals now, your muscular contractions, when you move your arm or you move your leg, is this energy that allows us to do that. Then also it enables the beating of cilia and flagella. Cilia are those tiny projections in your windpipe that allows the air to move up and down, or the food, especially the flagella in your intestines, allows during peristalsis for the food to move along the small and large intestine. Okay, so it also needs that energy for the cilia and flagella to move about. Then also it enables active transport. So inside the blood system, molecules are being transferred, or food molecules are being transferred from one organ to the other one, and for the molecules to go from the bloodstream, where there is sometimes there's a higher concentration inside the blood and lower within the organs, then it moves from usually moves from high to low. That's inactive transport, but your active transport is when there is a low concentration and needs to be moved to a high concentration. So ATP is that energy that allows active transport from molecules to be moved from one area in the body to another through a membrane where there is a low concentration. So it needs energy to go from a low concentration to a high concentration. Yeah, anyway. So then lastly is your bioluminescence. Think of a firefly with a glowing butt. It also needs energy, needs to get energy from somewhere to allow this chemical reaction to happen, so that one can see the firefly lighting up. Okay, then there are actually a couple of factors that affect the rate of photosynthesis. Firstly, it is light intensity, then your temperature, and also carbon dioxide concentration. Okay, so if you have little light, the photosynthesis happens at a very slow pace. But the more you increase the amount of light, you see that the rate of photosynthesis at this end of the graph increases. Then at a stage, it will also plateau. It will it will flatline basically. So usually at 38% of light intensity, according to this source. But anyway, as soon as you increase the light intensity, the fact of the matter is the rate of photosynthesis increases. Okay, then with temperature, the same thing. The more you increase the amount of temperature, the higher the temperature goes, it decreases the rate of photosynthesis. And usually this have an optimum temperature between 25 and 35. But then as soon as you increase the heat again, the enzymes, as I mentioned previously within photosynthesis, they denature. And when they denature, this process of photosynthesis can't happen anymore. And as soon as they die out, the rate of photosynthesis also decreases. So with temperature, remember, there is an optimum temperature that allows the rate of photosynthesis to increase. Okay. Then thirdly, carbon dioxide concentrations is much the same as light intensity. The more you increase it, the more the rate of photosynthesis increases, and it also plateaus after a while. It will stay the same. Okay, then I would like to show you guys an experiment where they explain how light actually affects the rate of photosynthesis, and that you do need light for photosynthesis to happen, and also showing that the byproduct of photosynthesis is oxygen.
So this is quite practical photosynthesis. Now, I'm sure you already know the word and formula equation for this vital biological process. And this is, of course, when green plants use carbon dioxide and water in the presence of light energy to produce glucose and oxygen. Now, in this required practical, you need to investigate how various different factors could affect the rate of photosynthesis. And so we're going to look at how we can change light intensity and how that affects the rate in this particular practical. So we're going to use an aquatic plant. This is just common pondweed that you can buy from a pet shop or a garden centre. And we've got this in some water at the moment, just keeping it nice and fresh. So we're going to take a piece about this long and we're going to pop it into a boiling tube. Now, before I do that, I'm going to put some sodium hydrogen carbonate solution in the boiling tube, as this will encourage the pondweed to photosynthesize. Now, I've clamped my boiling tube so that you can see inside the tube nice and clearly, but in the school lab you may have used a test tube holder to hold your test tube, but I've just clamped mine today. So I've got a nice sized piece of pondweed here, and I'm going to pop it inside my sodium hydrogen carbonate solution, and I'm going to make sure it's right below the level. And then, just to make it easier for us to see the oxygen as it's being produced, we're going to cut, while underwater, the stem of the pondweed. And then as we measure the rate of the photosynthesis, we're going to count the number of bubbles of oxygen gas we see leaving that cut stem. So I just need to get down here and use my dissecting scissors just to snip, there we go, the very end of my pondweed off. I'm just going to push that a little bit lower in my solution. There we go. So now I'm going to be able to count the number of bubbles of oxygen produced in 1 minute, and that will give us a measure of the rate of photosynthesis. So I've cut my pondweed at an angle across the stem because that's the best way of ensuring you get a nice steady stream of oxygen bubbles coming from it. So the independent variable is going to be our light intensity, and to change that we're going to change the distance between our pondweed and our light source. Now I'm using quite a powerful light bulb here, but actually it may be better, and you may have done this at school, to use an LED light source. This is because the LED sources don't give off very much heat energy, and of course because the temperature will also affect the rate of the photosynthesis, we don't want to have two variables changing at once because that would make the test unfair. But this is the one I'm using today, and so this is the one I'm going to investigate. So the end of my ruler I've moved in so it's directly in line with the bulb inside my lamp, and I've angled my tube with my pondweed in so it's aligned to the 10 centimetre mark on the ruler. So now there's a 10 centimetre distance between the light source and the pondweed. I can now turn on my light. Now I'd want to turn the overhead lights off at this point as well, so the only source of light on my pondweed is coming from my lamp directly. I'm not going to take my measurement straight away because the pondweed needs a little bit of time to adjust to the brightness of the light and start the photosynthesis at the correct rate. So I'm going to leave it just a few minutes and then I'm going to take my measurements. So I've waited a few minutes now and the pondweed has adjusted to the level of light that it's getting from the light bulb. And so now using my timer, I'm going to time for 1 minute and count the number of bubbles. And that's just the number of bubbles coming from the cut surface. There may be other bubbles coming from the leaves around it, but just to keep it a fair test, I'm going to count the ones from the end of the stem. So moving down to eye level so I get a really good clear view, off I go. So with the pondweed this distance from the light, we've got 23 bubbles in our first minute. So now I'm ready to change the value for our independent variable. So I'm going to move my pondweed away from my light so that it is now 20 centimetres from the light source. And again, as I did before, I'm going to leave it for a few minutes so it can adjust to the new level of light, and then I will count the bubbles again. Now I need to do 5 values for my independent variables, so I'll repeat the experiment with the pondweed at 30, then 40, and finally 50 centimetres away from the light source. And so your results will look a little bit like this. And as you can see, as the distance between the light source and the pondweed got greater, the number of bubbles produced in a minute decreased. There is another way that you may have seen this experiment being done, and this is where we actually collect the gas, because of course one of the errors that we may have introduced in the first method is that we've assumed that every bubble of oxygen produced is exactly the same volume. So you may see it where we collect the volume of oxygen so we can measure it more accurately, or you may just collect the oxygen to prove that it is oxygen gas that the pondweed is producing. So this method, I've got a large beaker full of water. I'm going to take some of my pondweed and I'm going to trap it under a glass funnel and place that in the bottom of the beaker. Now obviously again with the light source close by, the pondweed will start to photosynthesize. But to collect the oxygen, I need to take a boiling tube full of water, turn it upside down, and place that over the top of my funnel. And now, as the bubbles of oxygen are produced, rather than just disappearing into the air, they will pass into the boiling tube, displace the water, and then after a few hours, you should have collected enough oxygen that you can either measure its volume or you can test with a glowing splint, which should relight to prove that it's oxygen.
Okay, there you guys go. I hope you guys realized that the light was the independent source, like she mentioned, meaning you control it. It is not dependent on whether the plant will photosynthesize. The plant and photosynthesis is dependent on the light source. Okay, moving on. If my computer will work. Why is photosynthesis important? Okay, first of all, as we mentioned in the first slide, during photosynthesis, it uses carbon dioxide. And remember, carbon dioxide, we exhale carbon dioxide. So we don't want large amounts of it in the air around us, otherwise we can suffocate, we can die. So we want less carbon dioxide, it is used by the plant, and we gain oxygen from the plant when it photosynthesizes. Okay, so that is beneficial for us because we need to breathe in oxygen. Okay, so more oxygen and also more food. When this process happens, starch, like we mentioned, all the sugars and so on is made by the plant and is stored within the leaves. So think of the food web. These are your producers of food, primary producers. If it was grass, bugs would come. They are the primary consumers. They eat from the grass, and then your secondary consumers would be maybe, no shame, we can't eat lions. Something that will eat the bug. Let's say it's yeah, okay, fine, a mountain lion. It eats the bug, and then some people who do eat a mountain lion, we will be the tertiary consumers, and so on. Okay, so basically within the food web, these are the primary producers. They produce food to sustain life in the rest of the ecosystem. Okay, so all of these things, and also I mentioned less CO2, all of this equals sustaining life. So without photosynthesis, we actually can't survive. We need the oxygen, we need less carbon dioxide, and we need food. So it is vital actually for life on Earth. Then when we look at crop yields, we need photosynthesis because when we plant crops and things to produce food, photosynthesis can be manipulated to improve the amount of food that is produced within the plants. So a way to improve crop yields is to use greenhouses, as mentioned here. So how a greenhouse works is they use glass panels to allow sunlight to reach the plants inside. But then as soon as the sun radiation reflects, it struggles to go out. And let's go to, it says 1, 2, 3, 4, 5 radiation channels that wants to go back out, but only 3 comes out. So some of it is trapped back into the greenhouse, the infrared rays. It can be radiated back because of the glass, is reflected back. So there's the heat inside, the temperature inside of the greenhouse is much higher, maybe with a few degrees higher than the outside temperature. So the temperature inside increases. And we saw that as soon as the temperature increases during photosynthesis, it can also increase the rate of photosynthesis. But it needs to be regulated, because remember, if it goes over a certain temperature, like 35 degrees, it will actually decrease the rate because the enzymes die inside of the plant. So the temperature needs to be regulated. Another way how greenhouses can help is if you want to increase the amount of carbon dioxide inside of it, it will also increase the rate of photosynthesis. Sometimes farmers use gas cylinders, these CO2 cylinders, that will enable more carbon dioxide to enter the greenhouse. So that can be regulated within this confined space. Then also the amount of water you use. With sprinkler systems and so on, they try to make it as efficient as possible. They want to use the less amount of water because we don't want to waste water, and it's expensive if you use too much water. So inside a greenhouse, less water is needed, but the right amount is given to the plants to increase the yield that you get from the crops. Then also within this controlled space, we can reduce the amount of pests, because so many things like cutworms and things, they eat the plants on the outside, and we have to use pesticides and things that can also increase the value of the plants and also make them harmful for humans to eat. So within this confined space and controlled area, we don't need to use pesticides, and we reduce the amount of pests in general that can eat the crops. Okay. So in a nutshell, that is what photosynthesis is about and why we need it and how it actually feeds all of us.